Hey everyone, welcome to our first lesson on genetics. And I'm just going to remind you, based on the email, if you haven't done these things yet, to do those first before proceeding with the notes. So that would be complete assignment number 1 here, which is an introduction to Mendel's laws of genetics. And you're going to be reading the first few pages of your hard copy textbook that will help you through some of the vocab and terminology. And then once you're done that, then I suggested that you watch the crash course video on Mendel's experiments, as well as number 3, the TED Ed on Mendel's studies. All right, so if you've done that, then you're ready for these notes. I'm going to just remind you, it's really important to go through all that stuff. There's an enormous amount of vocab and terminology here that's going to be really important. And if you haven't done all that, and if you're not good with the vocab, this is going to seem really confusing. Okay, so Mendel's laws, these laws developed by from Mendel's experiments still govern many of the patterns that we see in genetics today. I mean, they haven't disappeared. Um, and so we're just going to kind of go through them, and we're going to add some details that Mendel didn't really know about as he was carrying out his experiments. He didn't know about DNA, he didn't know about genes and alleles. So we're going to kind of blend Mendel's terminology into our modern terminology to make sense of his experiments and what Mendel's laws show. Okay, so first off, one of the first patterns of inheritance discovered by Mendel is that of complete dominance. All right, and so in Mendel's early experiments, what we call genes today, he called factors. Right? He didn't know. He just knew there was these things that were being passed on from parent to offspring, and today, of course, we call that our DNA. We call that our genes specifically. And so, one of the things that's really important is that for every gene, there exists 2 alleles in an individual. All right. And so here we get into the vocab already. So when we're talking about alleles are different forms of a particular gene. And an individual that is the result of sexual reproduction can only possess 2 alleles, right? Remember that our chromosomes come in homologous pairs. We inherit one set of chromosomes from mom, one set of chromosomes from dad, and it doesn't matter what type of organism you are. If you're an organism that is produced through sexual reproduction, then you inherit one set of chromosomes from mom, one set of chromosomes from dad. And so in the case of Mendel's pea plants here, um, over here on the side, we're basically summarizing Mendel's experiments. And so there's sort of 3 major experiments, but we're going to look at just number 1 and then essentially number 2 here in terms of how he came up with this idea of complete dominance. All right, so first off, what we're looking at here is the specific trait for seed color. So if you remember through the videos, pea plants have several different traits and characteristics. Mendel proved this law of complete dominance with all of those different traits, whether it was height of the plant, whether it was seed color, whether it was pod color or seed shape. There's a variety of different traits. We're just looking at that for seed color to show his first experiment. And so what Mendel did essentially was he took a pure breeding yellow, um, a plant that was pure breeding for yellow seeds, and he crossed that with a plant that was pure breeding for green seeds. All right, and so just maybe I'll try and highlight that in yellow. I don't know if it'll show up, but There we go. So what we know is the gene for seed color is represented by the letter Y. Capital Y for yellow and lowercase y for green. All right. And Mendel maybe didn't use these letters. Mendel didn't know about alleles. He didn't know about genes. He just said there was this yellow factor and there was this green factor. But what he could do was he could produce pure breeding plants. That means that he could take two yellow plants, and if they always produced plants with yellow seeds, then he knew those parents to be true breeding for yellow. And so therefore we can show that as that individual parent plant as being big Y, big Y. Remember, two alleles for each individual because you're inheriting one chromosome from mom and one chromosome from dad. In this case, this plant inherited both yellow genes or yellow alleles for seed color. So therefore it's true breeding. Similarly, the green plant, which would be lowercase y, and again, he could show that plants of this nature, if they were true breeding, they would always produce plants that had green seeds. So his goal was to look at what happens when you cross a pure breeding yellow producing seed plant with a pure breeding green seed plant. And so what he found was that 100% of the F1 generation, and here we get into terminology again, right? So this F1 stands for first filial generation. And so again, he didn't know about what this genetic makeup was, right? But what he did know is that 100% of the offspring produced yellow seeds. So his question, right, was where did this green seed factor go? What happened to it? When he first designed the experiment and he was thinking, I've got yellow producing seed plants, I've got green producing seed plants, he had any variety of hypotheses that could have been produced. One is, well, I will get some yellow and I'll get some green. He thought, well, maybe I'll get some yellow, some green, and maybe a blend. Like when we mix green and yellow, we get blue. But he had some ideas of what he would get, none of which involved getting 100% yellow producing seed plants. So in his mind, this green factor had disappeared. So then he carried out a second experiment. And in his second experiment, this involved crossing the F1 generation. So what this meant is he took this F1, which in the case of all the offspring would have been like crossing a plant that was, again, terminology, heterozygous for seed color. So by heterozygous, this means that you're producing or that you have one of each allele. Remember, these guys up here, these guys, we would call them homozygous because they possess two of the same alleles, but all the F1 are what we call heterozygous. They have or contain one of each type of allele. So Mendel was essentially crossing a heterozygous plant for yellow seed color with another heterozygous plant for yellow seed color. These were, in fact, the F1 generation, right? And so again, he doesn't use these letters. He doesn't know about one being homozygous, one being heterozygous. He just crossed the two F1 plants. And what he got was something that looked like this. Again, he didn't see these different genetic makeups. He just saw that essentially three out of every 4, right? Or a 3-to-1 ratio of yellow seeded plants, yellow seed to green seed. And suddenly he sees this green trait in seed color reappear in a very specific ratio of 3-to-1. All right. So the green trait, although absent in F1, had reappeared in F2. Conclusion: this is his first law of genetics. It is a pattern of complete dominance in certain traits. In the case of pea plants, all traits work this way, but as we start to examine different traits in different organisms, there are many different patterns of inheritance, but Mendel discovered the law of complete dominance, which exists in lots of organisms, his pea plants included. All right. And ultimately, what it's saying by this complete dominance is we've got these 2 different forms of seed color. A capital Y we use for the color yellow, and a lowercase y we use for the color green. And that's to show that in all the F1, there was only yellow. And so Mendel hypothesized that in order to have the green reappear, the F1 must have possessed this green factor, but it was masked by the presence of the yellow factor, and thus complete dominance. Now, there was a third experiment that Mendel developed, but we're going to get into that after, and we're going to talk about sort of how we use these, how we set up genetics problems, how we try to solve genetics problems and try to understand patterns of inheritance by looking at these things called Punnett squares, okay? For now, I'm just going to briefly describe his third experiment, and we're going to come back to it several times. And part of this experiment was for Mendel to develop a deeper understanding of hereditary traits and how traits were passed on. So his first set of experiments simply just looked at one trait at a time. Maybe it was the height of the plant, maybe it was the color of the seed, or maybe it was the color of the flowers, but he was looking at just one trait at a time. In his third experiment, he started to examine 2 traits at the same time. So what he did in his F1 or in his first cross, again, he looked at pure breeding plants for what are round yellow seeds, so 2 dominant traits, crossed with pure breeding plants that were green wrinkled seeds. So there's 2 different traits here. There's the trait of the color of the seed, and then there's the trait of the shape of the seed. So color yellow or green, and shape round or wrinkled. And again, he's already discovered that round and yellow are dominant through his first set of experiments, and he's understood that green and wrinkled are recessive. Again, vocab, right? And so what he sees in his first cross, or his first generation, F1, is that they're all round yellow, follows the same complete pattern of inheritance. Then he does the same thing and takes the F1 and crosses those. So he crosses a round yellow with another round yellow, but they are what we call dihybrids. They are heterozygous for both traits, okay? And what we need to start understanding at this point is how do we develop the possible gametes that are present in these individual plants? And that's what we're going to get to next. When Mendel does this cross of these dihybrids, he gets this distribution of offspring, which we're going to come back and look at. But for now, we're going to move on and talk about some of the laws that Mendel developed from his early experiments. And then we're going to get into this aspect of Punnett squares in the next lesson. So to finish off, Mendel's laws, he developed the law of segregation from his first experiments. What this law of segregation is very much what you've learned from meiosis in chapter 6. And that is, when we examine the genetic makeup of an individual, each individual has two alleles. All right, again, Mendel would have referred to these as factors, but we now know these to be alleles. Each individual has two alleles for a specific trait because we inherit one allele from mom, right? We're going to inherit one chromosome set from mom, and we're going to inherit one chromosome set from dad. So if we're talking about a specific gene, we're going to have two factors for any given gene. These alleles, and this is where Mendel came up with his law of complete dominance. In order for this law of complete dominance to work, these alleles or factors segregate or separate during the formation of gametes. And remember, gametes is just the technical term for our sex cells, like our sperm and egg. All right. And so over here, we've got that review of meiosis. All right. And so what's being shown is just two pairs of chromosomes, so 4 chromosomes. But we can see that as those chromosomes line up, there's two different ways in which the chromosomes can line up during metaphase. In the first scenario, scenario number 1, let's say if dads were blue and mums were red, well, dads seem to have both lined up on the left side and mums have lined up on the right side. And this is going to create the following gametes down below. Either sperm that are carrying both dad's chromosomes or sperm that are carrying both of what would have been his mom's chromosomes. On the other side, if the chromosomes line up in a different way, here they're mixed. There's one chromosome from mom and one chromosome from dad on each side. And so when they line up in this way, we get a different set of potential sperm. All right. And these different gametes are created through this law of independent assortment. How these chromosomes line up during metaphase will create a variety of different gametes, which will create variation in our offspring. So each gamete will contain only one allele. So if we remember, the whole point of meiosis is the chromosome number goes from 2n to 1n. If your chromosome number is 23, like in us, that means your normal cell, 2 times 23, starts out with 46, but your gamete will have half the number, 23, right? That's because our homologous chromosomes separate during meiosis, creating gametes that are haploid, right? Another term. This cell starts off as diploid, but produces gametes that are haploid, containing only one allele for each trait, right? Now, keeping in mind that fertilization, if this sperm, let's say this one right here, happens to then carry its genetic information to an egg in which it fertilizes that egg, it is also, so I've drawn one big chromosome, one small chromosome, so we have this 1n haploid sperm and we have a 1n haploid egg, will produce a new individual which will have two alleles restored, right? And so we now produce the zygote through fertilization, in which you will have two of each chromosome restored and the diploid number restored. However, the genetic makeup is not necessarily the same as the parent based on the various types of alleles that can be produced. All right, so this is the law of segregation. Um, and the second law developed by Mendel was from experiment 3, the dihybrid cross. And this is the law of independent assortment, which, much like the picture shown above, right, is that during metaphase of meiosis 1, homologous chromosomes line up independently and separate independently regardless of how other homologous pairs line up. And all possible combination of alleles can occur in the gametes. So let's look at this example down here of an individual considering two different traits. So, oops, sorry about that. So we could say, if this is a pea plant, we could say T is for tall, right? And little t is the allele for short. So we have one gene that is governing height. And let's say A is for axial, right? In which flowers are present in the axial position. You'll have to recall the video in which that was one of the characteristics of his pea plants, and lowercase non-axial, right? Or terminal position of the flowers, position of flowers. So if we're talking about an individual that is heterozygous for those two traits, they're going to possess one of each allele. But what are the possible gametes? What are the possible gametes that this individual could produce? So I had to move my screen down, so I'm not sure what got cut off there. But down at the bottom, we have this question: what are the possible gametes that could be produced? If you want to pause the video and try and think of it on your own, and then start the video to see the answer. Okay, so if we look here, right, as this individual produces gametes, each gamete can only have one letter from each trait. That means a gamete can only possess one T and one A. So what are all the possible combinations? Well, this individual could produce gametes that are capital T, capital A, could produce gametes that are capital T, little a. And if we look at lowercase t with a capital A, or lowercase t with a lowercase a. So that individual can produce 4 gametes. That's it for this one. And next up is looking at how we then put these gamete formations into understanding the potential outcomes of offspring.